The overall goal of this procedure is to use a comparative species approach to learn more about the brain areas and neurochemicals associated with paternal behavior. The first step is to identify two closely related species that differ significantly in the expression of the behavior of interest. Next, both species are allowed to interact with pups and observed for behavior within the parental response ethogram. These behaviors are videotaped for further analysis. The next step of the procedure is to collect brain samples and analyze them for the presence of vasopressin immunoreactive tissue in the target area. Neuroimaging software is then used to determine species-specific differences in targeted brain areas. Ultimately, results can be obtained that show species-related differences in paternal responses and vasopressin immunoreactivity in specific brain areas.
The primary advantage of using the comparative species approach, as opposed to other existing methods such as pharmacologically manipulated or genetically engineered species, is that it allows you to utilize naturally occurring differences between closely related species as opposed to using more artificially induced behaviors.
Though this method can provide insight into paternal behaviors, it can also be applied to other behaviors, such as aggression, sexual behavior, or grooming.
Before testing, mice from two closely related species are separated into test groups of biological fathers, virgins with no parenting experience, and virgins with limited pup exposure. First, prepare the cage and the video recording system. Next, place a pup in the middle of the cage. Once everything is prepared,